Okay, well, a very warm welcome to you, Michael, and also to everybody else who's listening from all around the world. It's a great pleasure for you to join us for the University of East Anglia's May 2026 Lasdun lecture, featuring Professor Sir Michael Houghton. We're delighted to be joined by students, staff, alumni, and supporters from all over the world for this online-only event. My name is Professor David Maguire, and I have the privilege of being the Vice-Chancellor here at the University of East Anglia, and I'm speaking to you from the campus in Norwich. The Lasdun lectures are public lectures designed to highlight the special work and special people of the university. Today, we're blessed to have a contribution from one of our finest. But before we get to our speaker, let me tell you a little bit about how our alumni have been supporting the university. Our fundraising campaign is called Dare to Do Different. It represents the strength of our university community and the commitment that people feel to what happens here. Our alumni play a vital role in that story. They play a vital role long after they leave campus in some cases, because they continue to support our students, our teaching, and our future in many different ways. Today, we have the pleasure of hearing from one such alumnus, someone whose journey started at the University of East Anglia, and that led on to him being jointly named the 2020 Nobel Prize winner in Physiology or Medicine. Professor Sir Michael Houghton is a director of the Li Ka-shing Applied Virology Institute at the University of East Anglia, and is the Li Ka-shing Professor within the Department of Medical Microbiology and Immunology in the Faculty of Medicine and Dentistry. He is involved in developing vaccines against HCV and Group A streptococcus, as well as therapeutics for cytomegalovirus, Alzheimer's non-alcoholic fatty liver disease, and cancer oncolytics, as well as immunotherapies. Previously, he worked as a vice president for research in Novartis and Chiron Corporation in California, where he and Chiron colleagues, Dr. Keelin Chu and Dr. George Kuo, in collaboration with Dr. Bradley of the Center for Disease Control, discovered the hepatitis C virus in 1989, and then developed blood tests to prevent transmission of the virus from global blood supply. For this work, he was co-recipient of the Nobel Prize in Medicine in 2020, along with Dr. Harvey Alter of the National Institute of Health and Dr. Charles Rice of Rockefeller University. And he also received a knighthood from Queen Elizabeth II. Today, we are absolutely delighted to welcome Michael back virtually to the University of East Anglia, where he graduated in 1972 with a Bachelor of Science in Biological Sciences. Michael will speak to us, and then you will be invited to ask questions which we'll answer from you, the online audience. So that's all from me by way of introduction. I'm now delighted to hand over to Michael for his address.
Thank you very much, David. I'm sorry I couldn't be in Norwich in person. I'm right now in Hawaii, so I'm 11 hours behind you and have copious amounts of coffee to try to wake myself up. It is a great pleasure to interact with UEA. As David said, I graduated in 1972 with a degree in biological sciences. I was a mediocre student at the University of East Anglia, so I guess perhaps I can play some inspiration to students that have not yet found their way. I had a mediocre degree, but was able to develop a passion for medical research. And I think that's the key to career success. You've got to find out what your passion is. And once you found it, working hard is a pleasure. And I think it's a recipe for success. So I really want to talk to you today, not so much about the work that I did. I will address some of it. But I really wanted to talk about this theme on my title. I truly believe this century is going to herald a molecular medicine renaissance, a golden era of molecular medicine. It's because science and technology is growing really fast, exponentially. And I just think this is a wonderful time for students of molecular sciences and of clinical science to enter the field of molecular medicine. I wanted to introduce myself a little bit. So as you can see on this slide, I graduated from UEA in 1972. I spent 3 wonderful years at the U of A, basically growing up and trying to mature. I don't think I matured until the late 20s, but certainly UEA was a wonderful experience for me. Um, I then got a PhD from King's College, Biochemistry Department, University of London in 1977. I was really grateful to the UEA to award me an honorary Doctor of Science in 2019. It was a great pleasure to come back to the campus to receive that, and I especially enjoyed that because it was actually before I got the Nobel Prize in medicine in 2020. So I'm really grateful to the UEA to recognize my career. And as David said, in 2021, I got a knighthood from Queen Elizabeth II for services to medicine. Next slide, please. Thank you. So once again, just to introduce myself, what happened after I left UEA? Well, first of all, I was extremely sad to leave UEA. It was a 3-year course. I wished had been a 4-year or 5-year course. Excuse me. Um, but once I got over the shock of leaving this wonderful university with wonderful teachers and professors, tremendous mentors. I remember Dr. Michael Balls really was an inspiration. And also, I really enjoyed the teaching in other areas, physical chemistry, for example, and statistics. But anyway, I left and joined a company, actually. I joined the research division of a pharmaceutical company called GD Searle and Company. They were based in High Wycombe in Buckinghamshire. And I was really involved in basic science there. It wasn't really applied science to make drugs or vaccines, but there were some wonderful experienced people, scientists from around the UK, and I was a research investigator. After working there for 10 years, I became competent in the new area of recombinant DNA, uh, otherwise called as molecular cloning. From Stanford University in the States, this whole new field of being able to clone nucleic acids into bacteria was developed. And this was a wonderful advance in molecular biology. Dr. Paul Berg at Stanford was largely responsible with colleagues. So I found myself 10 years later in 82 at the forefront of this new technology, sometimes called genetic engineering. And I went west with my young wife and our 1-year-old son to California. And I joined a startup company called Chiron Corporation that had been founded by 2 professors at the University of California, Bill Rutter and Ed Penhote. And I became one of their senior scientists. Um, I spent 25 years at that company, and that is where my lab discovered the hepatitis C virus, which is what I'm best known for. And had a wonderful time, had a wonderful lab of people, and really enjoyed it. After 25 years, I felt I should do something different, so I joined an antiviral company in San Francisco called Epiphany Biosciences. I was their chief scientific officer for a couple of years. Like a lot of biotechs, they don't always work out, and this one didn't. Chiron certainly did, but this one didn't. So I moved on, and I was offered a chair at the University of Alberta in Edmonton, in Canada. A very nice chair. Canada had a plan, had a strategy to try to um, attract foreign scientists to Canada to boost their science and development. And so I joined the University of Alberta as what they call a Canada Excellence in Research Chair. It came with a lot of money, so there were two attractants for me, something new, as well as a lot of research funding, which scientists always need. Um, During my tenure at the University of Alberta, I'm still there, by the way. I joined in 2010. I still work there as a professor and director of the Li Ka-shing Applied Virology Institute at the University of Alberta. Um, it would be nice if it was at the University of East Anglia, David, but it is at the University of Alberta in Canada. And during my tenure there, we've set up a lot of biotech companies. One of them is called Aurora Vaccines. I'm the chief executive officer of that, and it's dedicated to developing a hepatitis C viral vaccine. Um, I also have other jobs. It seems as I get older, rather than getting a lower workload, I actually get an increased workload. But that's because I have a passion for medical research, and so to me, it's not work. Um, so I am the chief scientific officer for essentially a clinical research organization in Edmonton called API, Applied Pharmaceutical Innovation. And then, as if I wasn't busy enough, I took on a visiting chair professorship at Hong Kong Baptist University last year, but I'm officially a visiting chair as of this year, 2026. Next slide, please. So, David introduced me. My claim to fame. These are the highlights of what I've been involved with. The first it was in 1980, where I identified for the first time the structure of the human interferon beta gene. Human interferon is the basic antiviral response that our bodies make in response to viruses, but also bacterial infections. And so, using molecular biology, I was able to identify the structure of the gene and work on its characterization. Then at Chiron in California, I discovered the hepatitis D viral genome in 1986. It's a fascinating virus. It has strong similarities to plant viruses, of all things. So here is this human virus that actually requires hepatitis B to infect the liver. It cannot infect on its own, so it uses hepatitis B virus as a helper virus. But once the hepatitis D viral genome gets into the liver, it replicates at very high level and exacerbates liver disease. And so there are big efforts right now to try to control liver patients that are co-infected with hepatitis B and hepatitis D. And then my big contribution was, biggest, I should say, was discovery of the hepatitis C virus in my lab at the Chiron Corporation in California. That happened in 1989. That led to several things. We were able to develop blood tests that are used now all around the world. And whereas before the introduction of our blood tests, if you had a multi-unit blood transfusion to save your life, there was a chance of around 5%, 1 in 20, that you would leave the hospital with hepatitis C infection. Now, um, one of the greatest feelings is to have been able to develop blood tests where now post-transfusion hepatitis C is completely eliminated around the world in countries that use the test, which is most countries. Nearly all countries use the test now to defer blood donors that are positive for hep C. Then our knowledge of the viral genome, which we published in many papers, and it started a whole field of hepatitis C virology by many good virologists around the world. That led to major pharmaceutical companies developing curative antivirals. Now, if you have hepatitis C, it's important to get diagnosed, and then once diagnosed, to be treated with the modern antivirals that can cure patients within 1 to 2 months. They're very well tolerated, and that's it. The virus is gone. And then finally, I've been working for a long time on a vaccine to hepatitis C. We still need a vaccine. To eliminate an infectious disease, you need diagnostics, you need antivirals, but finally, you need a vaccine to eliminate the actual infectious pathogen from the planet. So I'm working hard now on that program, and my lab showed for the first time that it is possible to immunize non-human primates and protect them against hepatitis C. And this work has spanned many years. So when I started work in 1982, I was intending to work on interferons at Chiron Corporation, this biotech startup. But I was introduced to the problem of hepatitis C, and I decided with my experience from the UK working at Searle, I would have a chance of identifying this elusive infectious agent. So I just want to emphasize back in 1982, for the students in the audience, we had a very bare toolbox to try to identify a virus for the first time. Polymerase chain reaction had not yet been discovered. That was discovered by Kerry Mullis in the late 80s, and he ended up getting a Nobel Prize for that technique. There had been no antigen or antibody defined for hepatitis C. It was known as non-A, non-B hepatitis in those days, not hepatitis C until we identified it. But no antigen or antibody had been defined. If you contrast that with hepatitis B, where Baruch Blumberg identified what he called the Australia antigen, obviously in Australia, in Aboriginal populations, he thought it was a polymorphism in a host protein, but actually it turned out to be the surface antigen of hepatitis B virus. Baruch Blumberg was the recipient of the Nobel Prize for that work. Nor did we have a cell culture system. You really, ideally, to identify an infectious agent, you want to be able to grow it in cell culture. That was not possible for non-A, non-B hepatitis. Unlike the case with HIV, where at the Institut Pasteur in Paris, work by Luc Montagnier and Francois Bar, that's a misspelling, I'm sorry, Francois Barré-Sinoussi, they identified the HIV virus by growing T cells from patients with AIDS in culture and seeing in the electron microscope a retrovirus, which later was identified as the cause of human immunodeficiency virus. We had no high throughput sequencing in 82. It took a month to sequence 100 base pairs. Now, our genomes, our entire genomes can be sequenced in a few days with deep sequencing. And that was based on Frederick Sanger's work at the University of Cambridge, who really pioneered DNA sequencing. He got 2 Nobel Prizes, a wonderful scientist. So in other words, we had no molecular handle available. So for a molecular biologist, in some ways, it was a horror story. How do you go about finding a virus when you don't have any molecular handles? Next slide shows how we did it, please. Thank you. So I worked for 7 years before we identified hepatitis C virus. And what we did is it wasn't even sure it was a virus at that point. Some people were speculating it was a prion, which is an infectious protein, not actually an infectious virus. But we assumed it was a virus. we didn't know what kind of virus. Was it an RNA virus, a DNA virus? We didn't know its size. So I took chimpanzee plasma. Many groups around the world had shown you can passage what was then known as non-A, non-B hepatitis from humans into chimpanzees. And it was the only model, animal model, and it still remains today the only immunocompetent animal model. Um, so I took chimpanzee plasma that I got from the Centers for Disease Control, and I ultracentrifuged so hard that any virus, the smallest virus known to man, would be pelleted. In other words, would end up on the bottom of the tube. So I gave it a real hard centrifugal force. I then took the pellet, extracted both RNA and DNA because I didn't know what kind of virus it was. And then using reverse transcriptase, which was another invention from a Nobel laureate, Dr. Tiemann, I used reverse transcriptase in vitro to transcribe the RNA into DNA. And along with the DNA from the pellet, I was able to clone it into bacteria. So what happens is you end up with millions and millions of bacterial clones on an agar plate. Each one has a single nucleic acid that originated from the nucleic acids in the pellet from the ultracentrifugation. Furthermore, this was a bacterial expression vector called lambda GT11, developed by Stanford University researchers. So not only was it a great cloning vector, you could also induce the bacteria to express the single nucleic acid that it contained from the chimpanzee plasma pellet. What you end up then with is what's really, it was an early proteomics experiment. You've got millions of clones, each expressing their individual proteins from the chimpanzee plasma. And then you can actually interrogate those millions of clones by incubating them with serum from patients diagnosed clinically with non-A, non-B hepatitis. We assumed that there would be antibodies to the elusive virus, and by incubating the serum with these millions of clones, we were looking for an antibody antigen reaction, and we use radioactivity to detect it, and you get positive clones. Well, some of those clones turned out to be encoded by the chimpanzee genome, and that is not to be expected for an infectious agent. It should be a gene from a virus or a bacterium, which is not part of the human genome. So eventually, after 7 years, we found a clone 511, and we were able to prove in an unusual way that it really was derived from the hepatitis C virus. Why? Because we showed it was extra chromosomal. It wasn't from the chimpanzee or human genome. However, it was derived from a large RNA molecule of around 10,000 nucleotides, which is a typical size for viral RNA genomes. And furthermore, that RNA was only found in samples from chimpanzees and humans that had been infected with non-A, non-B hepatitis virus. And then finally, 511 encoded a protein that bound antibodies found only in non-A, non-B infections of humans and chimpanzees. So we did not use the traditional Koch postulates to prove that this was truly the infectious etiologic agent of non-A, non-B. We didn't need to, frankly. And that's how we identified hepatitis C virus. Next slide, please. We is reflects the gentleman on this slide. My colleagues were Quilliam Chu, George Kuo, who I worked with at Chiron, and also Daniel Bradley was at the Centers for Disease Control in Phoenix and then Atlanta. who gave us well, we worked with him to develop the chimpanzee plasma that we cloned from. And so this was definitely a team effort. Big projects require a team to be successful. It's very important. I emphasize that to students. And unfortunately, I was greatly honored, of course, to get the Nobel Prize in medicine in 2020. But it was unfortunate that only myself, as the project leader, was awarded the Nobel Prize. My three colleagues who worked with me for many years deserved this prize as well. And I look forward to the time when the Nobel Prize Foundation become more inclusive and less exclusive. They have a rule where only three people can get it. My other two colleagues that were awarded the Nobel Prize, as David said, were Dr. Rice from Rockefeller and Dr. Alter from NIH. Those two gentlemen played an important role, a very important role in the development of the non-A, non-B hepatitis field and the hepatitis C field. But the actual discovery of the virus was done with these three colleagues and myself. And there are many Nobel Prizes that are given which exclude key contributors. And this was another case of that. So I really look forward to the day when the Nobel Foundation modifies its selection criteria, because other prizes around the world mimic the Nobel in only awarding it to three people. And I see absolutely no rationale and logic in doing that. What is the difference between giving it to three people as opposed to giving it to half a dozen people? If they all made a major contribution, this is only the right and the fair thing to do. So I really hope the Nobel Foundation changes. There are many examples of great scientists being left off Nobel Prizes because the Nobel Foundation has a rule of three, no more than three. Okay, next slide, please. So it took us 7 years. By the way, we were doing a lot of other strategies, trying to find the cause of non-A, non-B hepatitis. We weren't just using that method that I indicated to be the successful one. But if we were doing it now, discovering hepatitis C virus would only take 7 weeks, not 7 years. And that's because there's been tremendous advances in deep nucleic acid sequencing. As I mentioned, in 82, it took a month to sequence 100 nucleotides. Now we can sequence our entire chromosomes in just a few days. You can send your blood sample to companies, and the following week they will send you your entire sequence with its interpretation of relationship to other species, including Neanderthals. So this revolution in nucleic acid sequence, combined with computational bioinformatics, as we saw with COVID, it was possible to identify COVID using high throughput sequencing and bioinformatics in a supercomputer to identify that virus. It took more than 7 weeks. It took maybe a few months. And there were a few false identifications of viruses as being the cause, but they weren't. So it took several months. But the point is, these days, you take a sample which you know is transmitting an infectious disease, you can do deep sequencing, and then you ask the supercomputer to tell you which sequences are tightly associated with infectious samples. So you obviously have to get a lot of samples sequenced. And then the computer will tell you which ones are specific to the infectious sample from a patient with the disease. It's a tremendously powerful technique, as we all know, and as we all saw for COVID. And this is the way pathogens can now be discovered. I was reading this morning that hantavirus, there's been an outbreak of hantavirus, a very dangerous virus on a cruise ship that was cruising to the Antarctica. And actually, one of UEA's professors, Professor Hunter, has been involved in this work and made a very important contribution. So, hantavirus was already known, has been known, I think, from the Korean War. That's where it first emerged in soldiers in South Korea. Um, but had it not been known, those samples from the cruise ship could have been subjected to deep sequencing and probably were, by the way. And using bioinformatics in a supercomputer, the hantavirus could have been identified for the first time. So it's a tremendous new technique. Next slide, please. So we discovered the virus in 1989. It wasn't until 2015, which is 26 years, before the discovery of very potent HCV antivirals was developed. It took the field working together, many universities, many academics, and many pharmaceutical companies worked together to produce a cocktail of HCV antivirals that very specifically targeted the replication cycle of the hepatitis C virus. Gilead, together with Arbutus, these are two companies in the US, they developed the first curative HCV antivirals. And in phase 3 clinical trials, which involved many clinical investigators, the leads were Justin Feld in Canada, in Toronto, and Dr. Foster in Australia. They treated patients, hepatitis C patients, now they could be diagnosed with these curative antivirals. And what they found is the cure rate. On the left, the y-axis is indicating sustained viral response, which essentially is cure rate. You can see that irrespective of the type of genotype of hep C, it's a very heterogeneous RNA virus. Excuse me. Irrespective of the actual strain of virus, these curative antivirals essentially enacted a cure in almost 100% of patients. You will notice that genotype 3, the cure rate was 95%, which is very good. But that missing 5% has now been dealt with by these companies. They've developed slightly modified antivirals. Now, every hep C patient can be cured for life. The virus is gone. And so this was a milestone in HCV healthcare. And I think really it's important if anyone has had a blood transfusion in their lifetime, um, and also generally baby boomers who lived through the 60s and 70s, where many people experimented with recreational intravenous drugs and might have shared needles. It's a good, it's recommended to get tested for hep C because, excuse me, this virus hides in the liver for many years, and people often are not aware that they have a viral infection. But actually, all that time, the liver's degrading due to the replication and the immune response. And it's important to get cured before it's too late, before the onset of liver cirrhosis and also liver cancer. So, baby boomers, anyone that had a blood transfusion is recommended to get tested for hep C. Then you can get on the antivirals and be cured within 1 to 2 months. And the cure is very well tolerated, the treatment. Next slide, please. Yeah, so it took 26 years to develop curative hep C antivirals. Today, it would take less than half of this time. Why? Because once again, computational science is having a profound effect on infectious disease control in many ways. Most of you have heard of Google DeepMind, AlphaFold. This is Google is not far from, I'm in Hawaii now, but I live in California, and not far from where I live in California is Google. And they've come up with this amazing algorithm in the computer. If you feed in a primary amino acid sequence, which, as I indicated, is very easy to produce these days, the algorithm can predict the structure of the protein. Not just you start with the primary sequence, but Google DeepMind AlphaFold can tell you the secondary and, more importantly, the tertiary structure. That's very important because once you know the structure of the target protein that you're going after or target enzyme, you can also instruct the supercomputer to screen billions of drugs for fitting into the active site of that protein or enzyme. So drug screening can be done at a much higher scale now in supercomputers. Of course, once it predicts hits, you've got to confirm those in biological assays. But this is a wonderful new era of drug discovery using computational drug discovery. Whereas traditionally, pharmaceutical companies would screen maybe 500,000 drug structures in the wet lab, as we call it, in biological assays in order to find a hit. Now you can do it computationally. So AlphaFold is just an example of what it does. You put in a primary amino acid sequence from sequencing from the DNA or RNA. It predicts the structure of the protein, and then the supercomputer can get to work and tell you what drugs combine to the key areas of that protein. Next slide, please. And this is an example. Since being in University of Alberta in Canada in 2010, and I'm still there, we use computation to tell us which drugs are going to bind to our interested target proteins. And this is an example shown here. In red was a hepatitis C antiviral that was developed by a company. It turned out to be toxic and unfortunately damaged some people in clinical trials. And so there was a big effort to try to identify why. Why did it actually harm people? What we showed here, you're looking at the 3D structure of a cardiac ion channel that's called the Hurg ion channel. It's a potassium ion channel in our hearts that contributes to regulating heartbeat correctly. And we showed computationally that the molecule shown in red, it's a small molecule, it binds right into the cardiac ion channel pore. And that's what the supercomputer told us. And then we confirmed in biological assays that indeed it does inhibit this very key ion channel, the potassium Hurg ion channel. So it's just an example of how computation can be used, not just to tell you what new drugs you can develop into clinical trials, but also it can tell you some of the off-target hits that drugs tend to have. It's kind of rare for a drug not to have off-target side effects. Next slide, please. So as you all know, while all this was going on, mRNA technology was developed very successfully by NIH, Moderna, and BioNTech in Germany, and Pfizer in the US. And mRNA delivery really saved millions of lives during the COVID pandemic, many millions. It's a great new technology. There are new versions of this now developing, and one is self-amplifying RNA. And a slide describing what it is. You have an RNA molecule shown on the left-hand side. It's an RNA molecule. Of course, it encodes the antigen of interest. It could be the spike protein. I'm using it to encode hepatitis C envelope glycoproteins, but self-amplifying RNA encodes non-structural proteins, 1, 2, 3, 4, and what they do is they replicate the RNA in vivo. And so you get this incredible amplification of the input RNA. Furthermore, the replication complex that is made by non-structural proteins 1, 2, 3, 4 makes a subgenomic transcript of your antigen of interest. And so you get a twofer. You get this tremendous amplification of the RNA encoding your vaccine antigen. And what does that lead to? Next slide, please. The advantages of self-amplification RNA is you now need a much lower dose, 2 or 3 micrograms, even less. That makes it easier to produce during a pandemic. And secondly, it reduces the reactogenicity, or if you like, the toxicity. If you have to put in 100 micrograms of RNA with the lipid nanoparticles included, there tends to be some side effects. But if you can only put in a microgram or 2 micrograms, it means it will be less reactogenic, and that will lead to less vaccine hesitancy. So it has that advantage. Data from Japan in 2024, they were the first country to approve a self-amplifying RNA encoding COVID spike protein as a vaccine. And the data is very interesting. It shows that the antibody levels, while they're similar in titer to using mRNA, the antibodies last longer when they're made from self-amplifying RNA, which is very important for respiratory viruses like COVID, influenza, respiratory syncytial virus. So that could be a real advantage. It needs to be confirmed, but it could be a real advantage of self-amplifying RNA. Next slide, please. So, being a hepatitis C person primarily, I've asked the question, is self-amplifying RNA, does it confer an advantage over mRNA? And I've worked with a company in Canada, Cytiva, and my own company that I formed called Aurora Vaccines has been working with Cytiva. And in short, the answer is yes. Self-amplifying RNA is giving us better immune responses than mRNA. Shown on the next slide. So we're looking here at guinea pigs. 8 guinea pigs were immunized either with self-amplifying RNA shown in green. Please look at the green dots. Each dot is an animal, a guinea pig. And you can see that 8 out of 8 guinea pigs developed antibodies that could neutralize infectious virus extremely well. We got 100% neutralization. When you look at the mRNA encoding the same gene cassette, that's shown in purple on the far right. Once again, 8 animals. And you can see that the neutralizing antibody response is much lower. So this is an example of how self-amplifying RNA can really be another step forward after the wonderful mRNA phenomena developed during COVID. Next slide, please. And then also go to the next slide because I'm running out of time as usual. Yeah, so RNA technology is a huge area. It's going to be tremendously important pharmaceutically, not just for vaccines, for seasonal infectious diseases, but also pandemics of the future. And for sure, they're going to come. It's one thing we've learned. You always get a pandemic virus infection every 5 or 10 years. We will get others. So it's wonderful technology to deal with those very quickly, but also there are many genetic diseases due to faulty genes, mutations in genes, and these cause a lot of disease. So now with RNA technology, it's possible to encode the wild type protein without mutations to cure genetic disease. And we've seen an example of this already with sickle cell anemia, where administering an RNA encoding the wild type hemoglobin was curative for patients. So this is going to be a huge area. We need to make RNA lipid nanoparticles safer. We're doing that by using self-amplifying RNA to decrease the dose, but we're also looking at removing the pegylated lipid. There's four lipids that are used in a lipid nanoparticle. One of them is pegylated. It's got polyethylene glycol attached to it, which is what we put in our antifreezes, or used to in our automobiles. And a small proportion of people in the community are hyperallergic to polyethylene glycol. So I'm working with leaders in Canada who are making LNPs lacking the pegylated lipid, and we think these are going to be much safer formulations for human use. And there's other ways that we are working to make RNA safer as well. Next slide, please. Yeah, at the same time, there's tremendous advances in what's known generally as synthetic biology. That means a lot of different things. But for me, working on hepatitis C vaccine antigens, it means being able to make recombinant antigens, which have to be very correctly folded and have to be treated very carefully, not to denature them when you purify them for human use. It means we have ways to actually improve the production of recombinant antigens. And just an example of that is shown on the next slide. Hepatitis C is very diverse. It's more diverse than HIV, and there are 6 major genotypes. So a global vaccine has to be able to protect against all 6 of these basic genotypes. Each genotype has numerous subtypes. Next slide, please. So what we've shown is we can actually produce better recombinant antigens, proteins, using what is loosely referred to as synthetic biology. And this is what's known as a heat map. It's in guinea pigs again, and it's showing 8 guinea pigs that received our old protein, our old vaccine antigen shown in the top, and the new one shown in the bottom. So on the bottom axis, the x-axis, we're looking at neutralization of very diverse strains of HCV that are seen around the world. And where you see a red square, that means that guinea pig was making very strong neutralizing antibodies to that diverse strain. So these guinea pigs were immunized with a recombinant antigen from just one strain, But if you make it in the right way, you can see at a quick glance on the bottom panel that this antigen raises antibodies that can neutralize all of the world strains very well. In contrast, the earlier form of our vaccine antigens shown at the top, you can see that there's not nearly as many red squares. In other words, we're not nearly able to generate such strong neutralizing antibody responses. So synthetic biology, in a general sense, is moving fast and is able to generate much better proteins. Next slide, I've nearly finished. Other rapidly evolving technologies include, the next slide, please, the wonderful invention of CRISPR-Cas9's gene editing. Jennifer Doudna and Emmanuel Carpentier received the Nobel Prize in 2020 in chemistry. I received it in 2020 in medicine. These two scientists received it in chemistry, I believe. And what it is, it's an invention that enables now all scientists to be able to edit genes. And you can edit in a way to cleave sequences that are maybe disease causing, for example, viral genomes. And these days you can do it for DNA genomes of viruses as well as RNA. And of course you can also do it to inactivate our own genes that are associated with disease. Many genes in our bodies are associated with many different diseases, and you can actually inactivate those using CRISPR-Cas9. I don't have time to go into the mechanism, but essentially you use a guide RNA shown at the bottom of this cartoon, and it selectively binds to the gene of interest. And then the caspase, which is an enzyme complex that is added to the guide RNA, will selectively cleave that particular gene. It not only can inactivate disease-causing genes, but it can actually modify them as well, potentially to make them good genes rather than disease-causing bad genes. So this is a wonderful technique that's been developed recently. And finally, next slide. There's a whole new era of science in molecular medicine developing around epigenetic silencing of disease-causing genes. So this is a technology, not gene editing. It's not affecting transcriptional control or translational control in the ways we've traditionally seen it. This is the ability to go in with a targeted nucleic acid and a system to actually render parts of our chromosome inactive. Our chromosomes have active areas which are actively transcribed and translated, but we also have part of our genomes that are inactive. They are shut down by histones and other proteins that are methylated and don't allow transcription. Well, wouldn't it be perfect if we can actually engineer to do that against disease-causing genes, many of which cause cancer, but also against viral genomes, both DNA and RNA. And this is a huge area that is developing, and it's looking very promising. Hepatitis B affects millions of people around the world, and you have to be on antiviral therapy for all of your life if you are a Hep B carrier. But epigenetic silencing now is a method that really offers true hope to cure people of hepatitis B by inactivating the viral genome so it can't replicate and produce virus that causes disease. So the next slide is my conclusion, I believe. Yeah, my conclusions are, there's an incredible growth of science and technology that's taking place. And this should lead, in my opinion, to the curing and the effective treatment of most serious disease of humans and animals this century. It will obviously extend lifespan and improve qualities of life for patients and their families and friends. And I consider the 21st century to be a renaissance in molecular medicine. And there is never a better time for young scientists and clinicians to enter the molecular medicine field. And I think the 21st century will historically be viewed as a golden age of biomedicine. And if I can help define UEA scientists and clinicians in any way, I'm very happy to help. Thank you so much.
Thank you so much, Michael. That was really inspiring and lovely to hear you sharing your story. I think all of us will take great encouragement from your work, particularly I think students and early career researchers who will have heard your story and be able to think about what they need to do in their future careers that lie ahead of them. I like particularly the fact that you showed us that science is partly about inspiration, but it's also about persistence, it's about innovation, and also it's about experimentation, trying different things, but also being true to that laboratory method to check out and test your ideas. I was also struck by the fact that you mentioned the importance of teams in their many manifestations. I'm pleased to see that you spoke about the ethics of awards. Not all of us have had the opportunity to achieve awards, but it's great to hear from somebody who has, how you view things. If anybody listening in today in the audience has a question, you can type it into the Q&A. Perhaps I could begin with one, Michael, please. Early in your talk, you spoke about the development of vaccines and said that there have been some clinical trials in non-human primates. What do you think the immediate prospects are of a vaccine for human primates?
Yes, I really think a vaccine against hepatitis C virus is feasible, David. We've shown very clearly that when we immunize non-human primates, then we challenge them intravenously with the virus, we can prevent the persistence of virus. And that's the most important target for a hep C vaccine. We've got to prevent viral persistence because over the course of many years, with its smoldering in the liver, it can actually lead in some patients, not all patients, but in some patients, it leads to serious liver disease, which can be end stage. The liver just stops working and people die. Or it can also develop into liver cancer, which is hard to treat without a transplant. So, no, a hep C vaccine is very feasible. It used to irritate me, actually, 20 odd years ago or more. A lot of good scientists in a lot of good establishments around the world were saying, well, a hep C vaccine is impossible. There's so many different variants, so many different strains. It's worse than HIV and it won't be possible. But you should never be pessimistic. We now know that despite being a highly variable virus, the surface proteins of the virus, irrespective of the strain, have highly conserved regions which antibodies bind to and prevent infectivity. They prevent the virus getting inside liver cells. And so I kind of caution some of my colleagues, don't be so pessimistic when you write up your papers for publication. It does affect grant awarding committees. Oh, this is an impossible task. It's never going to work, so I'm not going to give you a grant. You don't know. You've got to work through it systematically before being pessimistic and also too optimistic. But absolutely, a hep C vaccine will appear. We're in 26. Certainly next decade, there will be a hepatitis C vaccine.
Thank you. We've got a question in from one attendee who said, you talked about having a bare toolbox back in 1982 when you started your search to identify hep C. Thinking about your toolbox for vaccinations and antivirals now, is it well stocked with modern equipment, or are there still big gaps in your toolbox?
Yeah, well, at the University of Alberta, I've been able to get a lot of funding from the province of Alberta. Essentially, provinces in Canada equate to states in the United States. So I've been able to get good funding from the province. I've been able to get good funding from the national government of Canada. So we are well equipped. We have deep sequencing at our fingertips. We have funding to work with leading RNA technology companies. We have all the equipment. So I think my message, is really biotech, as we all know, has been very successful at curing many diseases and preventing pandemic viruses. It's been very successful medically. It's also been very successful at stimulating economies of countries. And this is going to be even more so as we go through this century. And so it's really important for a country to invest in biotechnology. And a great example of that, I visited China recently and have associated with a university in Hong Kong. The energy in China, the funding that's going into the basic science, which is very important, but also in translational science, developing drugs and vaccines and diagnostics. There is so much money being poured into science and technology in China. It's a wonderful model for other countries. It will pay back, not just medically, but it will pay the countries back their investments economically. Just look at Google. Google computational algorithms to do drug discovery. I mean, it's a $1 billion industry, and there are obviously very good examples of that in the UK. One can quickly think of antibody technology that was developed at the University of Cambridge. But my message to national governments is biotech is not only going to cure disease for all of us, it's going to stimulate your economies. So invest in basic science and in stimulating companies to form and translate the scientific discovery into a medical product. It's going to pay off in the long term.
Thank you. Another question from one of the attendees. Are antivirals subject to similar issues of developed resistance as treatments targeting bacteria, or is an antiviral cure like the HCV cure effective permanently?
Yeah, great question. Um, Viruses like hepatitis C, they're RNA viruses. When they replicate, they make a lot of mistakes. And they do that because when you make mutants, as we saw with COVID, you can get mutants that evade the natural immunity as well as vaccine immunity. And also, as they mutate, they can stop being sensitive to antivirals. Yes, absolutely. As in the case of bacterial antibiotics. So it's very important when you make an antiviral or an antibiotic that you do it in a way that targets an essential function of the pathogen. You target, for example, a protein that is absolutely essential for the viral life cycle. And then also you have a cocktail. Having one antiviral is not enough. You've got to target two or three different key points in the replication cycle. And when you do that, it makes it much harder for the virus to mutate, to evade the cocktail of antivirals. So the antivirals that have been developed by many companies now for Hep C, they are curative. Resistance is not a problem. With HIV, as many of you know, resistance is a problem. When a resistant viral mutant develops, we have other drugs that can treat the mutant. So that's how we can prolong the lives of HIV patients and they can live normal lives because of the large inventory of different antivirals. But with Hep C, these are curative. They have an antiviral targeting the polymerase of the virus. They have an antiviral targeting non-structural protein 5A. And that combination works tremendously well. There is no resistance against these drugs for Hep C.
Thank you very much. We have a potentially controversial question, but nonetheless important one, I think. President Trump's health secretary, R.F. Kennedy, and anti-vaxxers have obviously got their own views on work in this area. Has that impacted on your research capacity in any way?
Yes. Well, first of all, I'd like to apologize for, let me say, I did not vote for President Trump, and I disagree with virtually everything he's doing. And that includes the appointment of Kennedy as head of health and human services. I really don't understand what's going. Well, I do understand, but I'm appalled that United States, uh, which I was so proud to join such a vibrant medical community in 1982, and now I find I'm back in the caves making fires out of sticks, etc. It's appalling. How has it affected me? I do have to be careful because it's not just in the USA where there is this extreme right-wing politics. It does exist in Canada. So sometimes when I write grants, it might seem trivial to many of you, but I try and avoid the word vaccine. I I use the word preventative, and that could mean a drug or a vaccine. So yeah, sometimes I encounter similar psychologies in Canada, and I have to be diplomatic in how I approach it. But that said, I've received tremendous support from Canada's governments, both provincial and national. And I don't know what I can say. I think what it is, the COVID RNA vaccines saved millions of lives, but some people did experience toxicity. Young men, for example, developed inflammation of the heart. And of course, the other aspect of all this is the governments um, instructed all people to be vaccinated. So there's a lot of people that believe government should not instruct you to do anything, which I completely disagree with. In the interests of society, sometimes you have to do what the government instructs. It's for everyone's benefit. And we've seen that with the measles outbreak in the United States. It's in many states, measles is now causing severe disease in children. You have to have uh, a national or state edict that all children get vaccinated against measles. Otherwise, a lot of kids are going to die unnecessarily. So, how to explain it all? It's craziness, in my view, and hopefully we'll get into better times in the near future.
Yeah, we just have time for one last question. We've had a question in from a member of staff, actually. Someone has just recently graduated with a PhD and has obtained the first academic appointment and is wondering if you could offer any career advice to someone embarking on a career as to the balance between research and teaching, or just any guiding principles for starting out. What lessons have you got going all the way back to 1982?
Yeah, well, congratulations to that young person. Um, you know, I'm there are some outstanding teachers, and of course, our students at universities like UEA need to be taught very well. And an outstanding teacher, is doing a fine job. I'm not a good teacher myself, I admit that. But I've always been very passionate about medical research. And so I think if you're a passionate teacher, then um, continue. Make sure that you keep up with the field because molecular medicine is moving so rapidly. It's really hard to keep up with the field, but you got to keep up with the field and tell your students what all the new trends are, very exciting ones, like I've tried to do in this talk. If you're a researcher, what my advice is be passionate. I'm a medical researcher, not just to get appointments in different universities, not just to get grants, but my motivation is truly to make a contribution to human health. And I just think that's so important. If you're in the medical sphere, the molecular medicine sphere, be passionate about trying to make a difference to human health and their clinicians by giving them tools to better manage the patient. I have come across academics that seem obsessed with where they're going to get their next grant from, where are they going to get their next award from, which enables them to get another grant. And I do not recommend that. I recommend young academics in molecular medicine being committed to improving healthcare through their research. If you do that and you're passionate about it, you will be successful.
Thank you. Professor Sir Michael Houghton, thank you so very much for joining us today. It's been an absolute privilege to welcome you back to the university virtually, and we hope to welcome you back in person before too long. It's been an absolute inspiration for us to hear the little part that UEA played in your incredible career, and been wonderful to hear your story and your reflections and your thoughts on your field and the likely new development. So thank you very much indeed for joining us from Hawaii, and we wish you best rest of the day. Thank you once again from all of us here.
Thank you, David, and thank you very much to the University of East Anglia. You set me on my way for the career that I've described to you, and without having the teaching I got at UEA for 3 years, I would not be where I am now. And so, sincere thanks to the university and to all of my many teachers who really affected my thinking and philosophy. Thank you.